Hello, welcome to Learning Theatre. In today's video, we're going to talk about regulation of glycolysis. In our previous video, we did a detailed lecture on glycolysis pathway with all the substrate and their enzymes. So today, we are going to focus on three enzymes. That is the hexokinase, phosphofructokinase 1, and then pyruvate kinase. We are much focusing on these enzymes because they are much highly regulated and there are some hormones influencing them to control this glycolytic pathway. So this is our glucose. Let me do it here. Glucose. This is our glucose molecule. And this hexokinase, so this is our hexokinase enzyme. This is our hexokinase enzyme. Let me write it there. Hexokinase enzyme. So hexokinase. This hexokinase. So this hexokinase is what will convert this glucose to glucose 6 phosphate. And we have two types of regulation. And when we talk of regulation, we have two types of regulation. We have the allosteric and then the hormonal. And then an enzyme has a three-dimensional shape. It has an active site and then an allosteric site. The active site is what reacts with the substrate, and some factors will bind to the allosteric site to change the three-dimensional shape, and then this will change the activity of the active site, which will either enhance or inhibit it. And then the whole thing we're talking about is that the allosteric molecule regulators binds to the allosteric site to change a three-dimensional shape. And the hormonal has to do with the phosphorylation and then dephosphorylation. Yes, so allosteric allosteric molecule regulators is what binds to the allosteric site and then change the three-dimensional shape. And then the hormonal is what does the phosphorylation and then the dephosphorylation. Now let's talk about this hexokinase enzyme. Hexokinase enzyme. So what this hexokinase enzyme is doing is that it is converting glucose to glucose 6 phosphate. Whenever there are a lot of glucose, it will want to convert them to glucose 6 phosphate. So glucose is what is going to stimulate hexokinase and tell him that, hey, there are a lot of glucose, convert them to glucose 6 phosphate. So glucose will stimulate it and then it will convert those glucose to glucose 6 phosphate. So let me do that one. So this glucose will come and then stimulate hexokinase and say convert those glucose to glucose 6 phosphate. And also, if there are a lot of glucose 6 phosphate, um, glucose 6 phosphate will also tell hexokinase that, hey, there are a lot of glucose 6 phosphate, so stop converting glucose to glucose 6 phosphate. So glucose 6 phosphate also inhibit hexokinase from converting glucose to glucose 6 phosphate. So this is what we call the allosteric regulation. Allosteric regulation. When there are a lot of glucose, glucose stimulates hexokinase to convert the glucose to glucose 6 phosphate. And when there are a lot of glucose 6 phosphate, glucose 6 phosphate will also inhibit hexokinase from converting those glucose to glucose 6 phosphate. And with the hormonal regulation, we're going to talk about two types of hormone, the insulin and then glucagon. Insulin is released when blood glucose level are high. So insulin will break down the glucose to make ATP, and insulin will then go to the nucleus and then stimulate the synthesis of more glucokinase and then hexokinase, and this will increase the amount of um, glucokinase or hexokinase in order for them to convert all those glucose to glucose 6 phosphate. So whenever insulin increases the number of glucokinase and then hexokinase, the activity of glucokinase and then hexokinase increases. So insulin is released whenever we have a lot of blood glucose levels. And also, glucagon is when we have low blood glucose levels. So when we have low blood glucose level, glucagon is released, and then this is because we need glucose in the bloodstream. So whenever there's low blood glucose level, glucagon is released in order to help us maintain this glucose in the bloodstream. So this glucagon comes to inhibit hexokinase and then the glucokinase from continuing to convert glucose to glucose 6 phosphate. So this is the work of glucagon. It will come and then inhibit hexokinase and then the glucokinase to prevent them from still converting glucose to glucose 6 phosphate in order for us to get more glucose in the bloodstream. Now, the second type of enzyme we have is what we call phosphofructose kinase 1. Phosphofructose kinase 1. This one. Phosphofructose kinase 1. So let me make it PFK1. Yes. Phosphofructose kinase 1. This enzyme is what phosphorylate fructose 6 phosphate to fructose 1,6 base phosphate, and is one of the most highly regulated steps in glycolysis. This step of converting fructose 6 phosphate to fructose 1,6 base phosphate is the most highly regulated step in glycolysis. So, whenever you are making too much ATP through glycolysis, that is going to tell this enzyme here that there are a lot of energy supply in the body. So don't keep doing glycolysis. Whenever there is too much glycolysis or there is too much ATP through the glycolytic pathway, and that is going to tell this enzyme to stop the glycolytic pathway. So the first thing that inhibit this enzyme is too much ATP. So too much ATP inhibit phosphofructose kinase 1. So ATP. So ATP comes in and then allosterically inhibit phosphofructose kinase 1. So one inhibitor of phosphofructose kinase 1 is ATP. Another thing that can also inhibit phosphofructose kinase 1 is the Krebs cycle intermediate known as citrate. The Krebs cycle intermediate known as citrate. So a lot of citrate means there are a lot of Krebs cycle activity that is leading to the formation of a lot of ATPs. But we don't need too much ATP. So citrate will then come in and then bind and then inhibit the Krebs cycle activity. And whenever there are a lot of Krebs cycle activity in the Krebs cycle activity, that leads to the formation of ATPs, citrate comes in and then bind to the Krebs cycle activity to stop the Krebs cycle activity from continuing to make more ATPs. So citrate is an inhibitor. So citrate, yeah. And there is one that comes in to stimulate phosphofructose kinase 1. So whenever we have a lot of ATP being broken down to ADP and inorganic phosphate, the concentration of ATP goes down. That is a lot of ADP means low ATP. So more of ADP will stimulate phosphofructose kinase 1 to make more ATP. So whenever we have more ADPs, that is telling phosphofructose kinase 1 that it has to make more of ATPs. So ADP also stimulate phosphofructose kinase 1. Now, this one here is also two enzymes combined as one. This part here is known as phosphofructose kinase 2. Phosphofructose kinase 2. And then this one here, this part of this enzyme is known as fructose 2,6 bisphosphatase. Fructose 2,6 bisphosphatase. So, fructose 6 phosphate in the presence of phosphofructose kinase 2 can convert to fructose 2,6 bisphosphate. Yes, so fructose 6 phosphate in the presence of this enzyme can convert to fructose 2,6 bisphosphate. And then let me do this one before. So let me do it this way. Fructose 6 phosphate, in the presence of phosphofructose kinase 2 can convert to fructose 2,6 bisphosphate. Yes. And this fructose 2,6 bisphosphate can also bind to phosphofructose kinase 1 and stimulate it to make it active. So this one here, this one, fructose 2,6 bisphosphate can bind to phosphofructose kinase 1 to make it active, then so that it can convert more of fructose 6 phosphate into fructose 1,6 bisphosphate, which will lead to more ATP. Fructose 2,6 bisphosphate is a most powerful regulator of phosphofructose kinase 1. And the other domain of this enzyme, known as the fructose 2,6 bisphosphatase, convert this fructose 2,6 bisphosphate back to fructose 6 phosphate. Remember that this phosphofructose kinase 2 converted fructose 6 phosphate to fructose 2,6 bisphosphate, which stimulated phosphofructose kinase 1 to make more ATPs. And then this enzyme here, fructose 2,6 bisphosphatate, will come in and then convert this one, this fructose 2,6 base phosphate back to fructose 6 phosphate. So let me do this. Let me do this one here. This fructose 2,6 base phosphate in the presence of fructose 2,6 base phosphatate will be converted to fructose 6 phosphate. So fructose 6 phosphate. Yes. And we need to inhibit phosphofructose kinase 2. This phosphofructose kinase 2 in order and in order for us to do that, glucagon will do that. So that it can't convert fructose 6 phosphate to fructose 2,6 base phosphate again. In order for it to stimulate phosphofructose kinase 1. So in order for glucagon to inhibit phosphofructose kinase 2 to prevent it from converting fructose 6 phosphate to fructose 2,6 base phosphate, it will add a phosphate group to phosphofructose kinase 1 to inhibit it, and also add a phosphate to fructose 2,6 base phosphatate. So it also add a phosphate to this one here too, and stimulate this enzyme here. So a glucagon add a phosphate to fructose, a phosphofructose kinase 1 to inhibit it, and then also add a phosphate to fructose 2,6 base phosphatate to stimulate that one. It does this. When it does that, it stimulates fructose 2,6 base phosphatate to be able to convert fructose 2,6 base phosphate to fructose 6 phosphate and prevent phosphofructose kinase 1. So insulin comes in and then remove the phosphate that is on phosphofructose kinase 2 and then fructose 2,6 base phosphate. So, and then in doing that, this will stimulate phosphofructose kinase 2 and then inhibit fructose 2,6 base phosphatase. Now, let's look at the third enzyme. This is the third enzyme. And then this enzyme is known as pyruvate kinase. This enzyme generates ATPs by taking phosphate from phosphoenolpyruvate and add this phosphate to ADP to make more ATP. So when there is too much ATP in an acetyl-CoA, and then when there is long chain fatty acid, this long chain fatty acid means that a lot of beta oxidation. And when there's a lot of acetyl-CoA, this step is going to be inhibited. So glucagon will phosphorylate this enzyme to inhibit it to avoid too much breakdown of glucose. So there are so many things that inhibit this pyruvate kinase. Remember, I said that when there is too much of ATP, this enzyme will be inhibited. So one, when there is too much ATP, this enzyme is inhibited. And also when there is a long chain fatty acid or a lot of beta oxidation, so this one is also going to inhibit this enzyme. Long chain fatty acid or lot of beta oxidation inhibit pyruvate kinase. So that one too, let's do that one too. Long chain fatty acid, long chain fatty long chain fatty acid or beta oxidation. Let me add CoA to it. CoA. I added the CoA because fatty acid has a CoA group attached to them. But when we have a lot of acetyl CoA, this enzyme can also be inhibited. So let me do that one too. A lot of acetyl CoA. Acetyl CoA. Yeah. And also, I said glucagon will also phosphorylate this enzyme in order to inhibit it, to avoid it from making or from breaking down a lot of glucose. So glucagon also inhibit this enzyme to prevent it from a lot of glucose breakdown. So let me do that one too here. Glucagon. Yeah. And we also have those that will come and then stimulate this enzyme. And also the first one is insulin. Insulin will stimulate this enzyme and by increasing the synthesis of pyruvate kinase. So insulin does this by dephosphorylating the enzyme. Remember, this glucagon phosphorylated this enzyme to prevent it from continuing to break down glucose. And then insulin will come, and then in order for insulin to stimulate this enzyme, it has to dephosphorylate that phosphate that was added to it. So in doing that, it stimulates this enzyme. So let me do this one too. Insulin is a stimulator of pyruvate kinase. And the second one that will stimulate this enzyme is fructose 1,6 fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate stimulates this enzyme, and this kind of stimulation is known as feed-forward reaction. Feed-forward reaction. So this means that fructose 1,6 bisphosphate, it is up there, and then pyruvate kinase is down there. So fructose 1,6 bisphosphate is producing a lot or large amount of ATPs, and then it is sending a message to this pyruvate kinase and telling him that, Master, get ready, for I am making a lot of ATPs. So you better increase your activity in order to break down. So when fructose 1,6 bisphosphate stimulate pyruvate kinase, we call that feed forward reaction, meaning that fructose 1,6 bisphosphate is producing a large amount and then coming down to sending a signal to come and then tell pyruvate kinase to get ready or increase in its activity. So that is what we call feed forward reaction. So let me do that one too. So the second stimulator is fructose 1,6. Prince, thank you for watching this video. Please like it, comment on the comment section, and then subscribe this video for us, or subscribe to this channel, and then remember to press the notification bell and get notified whenever we upload a new video. Thank you for watching.